Hello everyone. We discuss about the next generation CRISPR technologies, and in these technologies we have talked about the base editing technology, the cytosine base editing technology and adenosine base technology. We talked about the dual base editing as well as the CBE based deletion technology that has been developed. Although these CBE and ABE can induce precise base transitions, the tools for generating base transversions are limited. So, another technology was developed in 2019 which allowed these CRISPR based technologies to be used to create prime editing as well as basically the precise editing. It increased the efficiency of editing, and now it could also be applied to a wider range of platforms. This prime editor technology, it produces in human cells all 12 kinds of base substitutions, precise insertions of up to 44 base pairs and deletions of up to 80 base pairs and combinations of all these edits. So, this prime editing, what does it contain? These prime editors, they uses a fusion of nickase Cas9. Now, recall what the nickase Cas9 is. Nickase Cas9 is a Cas9 which does not have a nuclease activity. So, its nuclease activity is impaired. Now, in the CBE and ABE, we talked about the D10A mutation to create the nickase Cas9. The prime editor uses different mutation, but again it generates a nickase Cas9. And along with this nickase Cas9, a reverse transcriptase is used. Now, reverse transcriptase, again recall that it converts the RNA back to DNA. This is a reverse transcriptase which reverses the mechanism of transcription, transcribing the RNA back to DNA. This fusion of nickase Cas9 and reverse transcriptase is complexed with a prime editing guide RNA which we call as the PEG RNA. So, we have talked about this sgRNA which is a single guide RNA. So, prime editing guide RNA is a PEG RNA. Now, PEG RNA is composed of a reverse transcriptase template and a primer binding site at the 3' end of the sgRNA. So, the sgRNA, it is a typical RNA which has the sequence for the target site because it is the guide RNA. Along with this, this also contains a reverse transcriptase template as well as a primer binding site at the 3' end of the PEG RNA. This reverse transcriptase templates contain the genetic information for the desired mutations, and the primer binding site pairs with the nickase Cas9 nicked single stranded DNA, priming the reverse transcription and incorporating the genetic info from the reverse transcriptase template into the genome. So, because it is using, so what does it do? Because we want the transversion to occur, we are providing an information on the reverse transcriptase template, whatever information we want to feed into the genome. So, whatever change we want to incorporate into the genome, we are sending it by the way of these reverse transcriptase template. So, this reverse transcript template contains the genetic information for whatever desired mutation we want. And this primer binding site which is adjacent to that, it pairs with the nickase Cas9 nicked single stranded DNA, priming the reverse transcription of incorporating the genetic info. This is then followed by equilibrium between the edited 3' flap and the unedited 5' flap, ligation and repair which generate the desired edit. So, we will see it in the figure that how this is happening. So, this is the kind of the CRISPR-Cas9 structure. It has a guide RNA. Now, this guide RNA also contains a reverse transcriptase template and is bound by a reverse transcriptase enzyme, and there is what is called as a primer binding site. So, this kind of a system has been developed for primer editing. So, what this primer editing will do is that this reverse transcriptase template with its primer binding site is going to bind here, and then whatever this nickase has created a nick. So it is going to generate two different ends, a 3' flap and a 5' flap. So for that the equilibrium will occur, the 3' flap and 5' flap, and it will cause the desired mutation to be incorporated into the genome. So this genome is now edited in this manner. So this is nicked, and there is a base pairing of the reverse transcriptase as well. So this is then followed by the equilibrium between the edited 3' flap and the unedited 5' flap, ligation and the repair, which generate the desired edit. As prime editor generates base substitutions and short indels at a relatively wide range of positions, ranging from +1 to +33, counting from the base 3' of the PEG RNA induced nick, it is not substantially constrained by its PAM. So this is again an additional advantage, that it is not restricted by the PAM, that it has to be very adjacent to the PAM, because it creates the substitutions on a wide range of positions from +1 to +33, counting from the first base of the 3' end of the PEG RNA induced nick. So this is the primer editing mechanism. So we are providing a primer as well in this case, the primer which is priming the reverse transcription of a template that we are giving, and this reverse transcription template is the kind of mutation that we want to be incorporated into the genome. So in the genome a nick will be created by the nickase Cas9, and the primer will cause the reverse transcription of the primer of the reverse transcriptase template, and then that information will be incorporated at the 3' end where the nick has been created by the PEG sgRNA, PEG RNA basically. So this is again a modified system and a very useful and efficient system to create the editing in the such as the base substitutions, transversions, etc. So this is all about the next generation CRISPR technologies. Many variants have occurred, and now as we say, newer and newer technologies have also been developed and adopted. But these are some of the very latest technologies that have been developed on the CRISPR-Cas9 platform. Now we have discussed about the basic CRISPR-Cas9 locus. We have discussed about the mechanism of action of CRISPR-Cas9. We have discussed about the next generation CRISPR-Cas9 based technologies. Because this is such an important gene editing tool, we have been doing in detail all the things that has been associated with it. So now we come to discuss about the applications of this CRISPR-Cas9, and again I will take multiple sessions to discuss about the applications of CRISPR-Cas9, because of its significance and its utility on various and wide platforms. Today I will discuss about the CRISPR-Cas9 applications in agriculture. So when we talk about this CRISPR edited agriculture, the development of genetically edited crops. So you must have heard of GM crops, which are the genetically modified crops. So here also the modifications occur, but they are many times referred to as genetically edited crops or GE crops, similar to those developed by the conventional or the mutation breeding using the CRISPR-Cas9 makes it a promising and extremely versatile tool for providing sustainable productive agriculture for better feeding of rapidly growing populations in a changing climate. So as you know, the food security is a very important topic, and it has been in fact one of the goals in our sustainable development goals, UN stated SDGs. There is a zero hunger thing, and so there has to be a food security, so that the food is provided, a good quality food is provided for rapidly growing population, And then there is also the changing climate, because the environment is changing, and then therefore the plants have to be resistant to all the changes, all the adverse changes that have been occurring in the climate. So we want those varieties, as well as we want the varieties which are resistant to diseases, which are resistant to pathogens. So we want these varieties to be developed for the sustainable productive agriculture. These genetically edited crops are very, very promising, and they have been developed using this CRISPR-Cas9 gene editing tool. These are some of the gene edited crop species. So as you can see, ranging from the cereal plants to the flowering plants or the ornamental plants, in fact all the crop species, various kind of crop species have been edited using the CRISPR-Cas9 mechanism. So legumes, staples, ornamental plants, herbs, all different crop species have been, the research is going on for developing the gene edited crops of these crop species. Primarily, there are three ways that the CRISPR is solving the world's food crisis, or what we call as crop improvement. In agriculture, we will talk about three different aspects. The first is the crop improvement aspect. The second is the domestication of wild species, and the third is the application in improving the breeding technologies only. The technologies, the biotechnology technologies for the breeding which are there, we can advance those also using employing the CRISPR-Cas9 mechanisms. So we first start with the crop improvement technologies, and there are three ways primarily to do that. First is by restoring the food supplies. What does that mean? That implies to increase the crop yield. So the crop yield needs to be increased so that there is no scarcity of food. There is food security, zero hunger, food for all. So that is the major application where the CRISPR gene editing is helping in crop improvement. Then it is also helping the plants to survive in hostile conditions. Now these hostile conditions can be stress, can be drought, can be flood, all different, can be, could be insects, could be pests, could be various kind of diseases. So any hostile condition, the conditions outside or within the plants that have been developed. So they have been developed because of the attack by the pathogens. The pathogens can be virus, bacteria, fungus or insects. So all these kind of hostility that has been done against the plants, these crop improvement technologies help the plants to become resistant to all these hostile changes. And again, CRISPR is solving this problem to a large extent. The third is improving the overall health of the plants. So, again, in overall health, when we say the crop quality as well. So the crop quality is also, the texture, the palatability, the nutrient constituents, all these things are also improved, which is implying the overall health of the plants. So these are the major three ways that we will discuss. So let's discuss, to start with, the approaches to increase the crop yield. Majorly, the approach to increase the crop yield is by manipulating the cytokinin homeostasis. So cytokinin helps in the growth or the foliage, these are basically the growth hormones. This kind of balancing, when you manipulate, then you can increase the yield. This has been done in Oryza sativa. The editing C-terminus of Oryza sativa, LOGL5, which encodes a cytokinin activation enzyme in rice, it enhanced the grain yield in a variety of environmental conditions. So one important aspect that I would like to mention here is that of course these CRISPR-Cas9 technologies are very promising, very efficient, and they have been employed to edit, as I said, basically many crop species, and in our subsequent sessions when we will discuss about the therapeutics, it has been promising in case of cancer, in case of various kind of diseases, animal diseases as well. But again, this is not a very old field. Therefore, the research and the trials have been going on, and before we it is employed or these are released into the market for consumption, obviously it takes time. So these are all the researches that is happening and have been promising. So for example, the editing the C terminus of Oryza sativa, it encodes the cytokinin activation enzyme in rice, and by editing that by using the CRISPR technology, it has shown the enhanced grain yield in variety of environmental conditions. Then another thing is knocking out the gene. Knocking out the gene that encodes cytokinin oxidases or dehydrogenases, an enzyme that catalyzes cytokine degradation, it also has shown high yield phenotype in wheat. So the cytokine degradase or the cytokinin activation has been shown to produce high yield in rice and in wheat. Similarly, another example, knocking out the gene that encodes amino acid permease 3, which is involved in nutrient partitioning. Rice varieties were bred with elevated tiller number and increased yields in combination with maintaining the grain quality. So the grain quality is maintained, however, the yields are also increased by encoding the amino acid permease 3, which is involved in nutrient partitioning. So whatever this rice is taking up, the nutrient, when that enzyme is knocked out, which is causing the nutrient partitioning to be there, then the increased yield has been there, and this has been knocked out by using the CRISPR-Cas9 technologies, employing the basic CRISPR-Cas9 technology or the technologies that we have discussed, the CB, AB, the dual ones, particularly the dual fusion ones, or the primer editing that we had discussed. Editing of other genes including Oryza sativa PN5B, which regulate the panicle size, Oryza sativa GS3 that regulate the grain size. So this is also, we will talk about in crop quality. So they are increasing the grain size. In Tritium aestivium GW2, Oryza sativa GW2 and Oryza sativa GW5, they are regulating the grain weight. They have also, these quality improvement has also given rise to crop plants with increased yields. So food security, food scarcity is very promisingly, there are researches which is showing promisingly efficient methods to increase the yields and combat these kind of scarcity that the nation or in fact the whole world is facing. The next is the approaches to design the disease resistant. So I am not taking all the different kinds, but let us take the disease resistant. There could be stress tolerance, there could be herbicide resistant plants, there could be insecticide resistant, pesticide resistant plants, there would be pathogen resistant plants. So different varieties when we are talking about the crop improvement, in case when it is helping the plants to combat all these kind of holistic environments, and in that we will take one example of, for example, the approaches to design the disease resistant plants. They are done by different mechanisms. The first is the gene disruption via indels in coding sequences. So insertions and deletions are generated in the coding sequences, and therefore the genes are disrupted that result in the disease resistant plants. It is used in several crop plants, including the essential cereals such as rice and wheat, to introduce the trait of interest. The resistance is engineered by disrupting a plant susceptibility gene. It is termed as a S gene, which alters the plant pathogen interaction, leading to the reduced pathogen fitness on the host plant. So, a pathogen is able to survive on the host plant or cause a disease because of the expression of the susceptibility gene. So, if this susceptibility gene is disrupted by using the CRISPR-Cas9 platform, then the resistance is generated that alters the plant pathogen interaction, leading to the reduced fitness of the pathogen and therefore preventing the disease formation. It has been done, for example, in the introduction of indels affecting eukaryotic translation initiation factor 4e proteins. It has successfully induced resistance against multiple RNA viruses in Arabidopsis and cucumber. So, it has been shown that multiple RNA viruses, it become resistant to that because of the indels created in this eukaryotic translation initiation factor. The next mechanism by which it can cause the resistance to the diseases is the gene disruption via indels in the promoter regions. So, this can be applied in two ways: to disrupt the promoter sequence, blocking the gene expression entirely, or to disrupt an effective binding site, disrupting the plant susceptibility by preventing a pathogen effector binding to the promoter. So, as you know that various genes are transcribed and the promoter is responsible for the transcription of the genes, gene disruption via indels in promoter regions. This can be applied in two ways: to disrupt the promoter sequences blocking gene expression entirely, or to disrupt an effector binding site, disrupting the plant susceptibility by preventing a pathogen effector binding to the promoter. So, again this will regulate how the pathogen interacts with the plant and cause the disease. For example, the modification of the promoter of the rice sugar transporter gene, breaking the connection with the effector from a bacterial blight pathogen and thus leading to blight resistance. So, this has been also done. Additionally, CRISPR alter the gene regulation by targeting upstream open reading frame regions and editing the cis regulatory elements. So, this CRISPR can edit the promoter sequences, again either blocking the entire gene, the susceptibility gene, therefore reducing the fitness of the pathogen, or altering the promoter effector regions, which are basically affecting the pathogen interaction with the plant, causing the disease. So, these are again the examples how this kind of things are done. The gene deletions are also done by multiplex sgRNAs. Here multiple sgRNAs are used to introduce multiple double-stranded breaks at precise locations in the target genome. So, multiple guide RNAs can be used with the one single Cas9, and it can introduce multiple double-stranded breaks at precise locations in the target genome. These DSBs result in the removal of the DNA fragment containing the gene of interest before the cellular repair NHEG machinery repairs the double-stranded breaks. So, before this non-homologous end joining repair mechanism become activated, the fragments are removed by creating double-stranded breaks at multiple points because of the use of the multiple guide RNAs. In S-gene clusters where multiple S-genes reside in adjacent chromosomal locations, entire chromosomal fragments can be deleted, which is likely to generate long-term resistance to the target pathogen. For example, this has been employed in wheat varieties resistant to powdery mildew. Now, powdery mildew is a fungal pathogen. It has been generated by knocking out the MLO, which is a PM susceptibility gene, simultaneously activating a dominant gene that fully restores growth and yield on wheat. So, once the pathogen gene has been inactivated, which prevents the powdery mildew fungus to attack the wheat variety, as well as a dormant gene, dormant gene and not a dominant, activating a dormant gene that fully restores the growth and yield in wheat. So, such kind of crop improvement have also been done. Another is that the gene insertion. So, these were like knocking out or mutating the genes for knocking out their function, but the gene insertions can also be done by homology directed repair mechanisms, and in this the resistance genes or the R genes can be inserted. A delivery fragment containing an R gene surrounded by the sequence homologous to the DSB ends can be created, which is supplemented with the Cas9 and the sgRNAs. This cassette then guides the HDR mediated insertion of the R gene between the two DSB sites. So, when the double strands are created, then R genes or the resistance genes can be incorporated, resulting in the gene edited or genetically modified plants which are resistant to a particular condition or a pathogen. For example, HDR mediated resistant gene targeting has been done in Arabidopsis, tobacco, soybean, tomato, rice, maize, wheat, potato, barley, flax and cotton. So, these kind of HDR mediated resistant gene targeted have also been done. Another mechanism is the biomimicking. Biomimicking via promoter, allele or gene replacement. Biomimicking refers here to the introduction of CRISPR mediated mutation in such a way that the sequence of the target gene is converted to the sequence from a disease resistant variety. So, mimicking is basically imitating, and biomimicking because it is imitating a gene. So, disease resistant variety or disease resistant gene, this CRISPR mutated mutation is done in a gene that mimics the disease resistant variety. For example, the introduction of the N176K substitution encoded by the virus resistant allele of this effector molecule in Pisum sativum into the Arabidopsis elongation initiation factor gene to generate Arabidopsis plants resistant to clover yellow vein virus. So, the elongation factor substitution which is a virus resistant allele, this N176K, this substitution is encoded by this particular allele. When this is incorporated into the Pisum of the Pisum sativum is incorporated into Arabidopsis in elongation initiation factor gene, then it generates the Arabidopsis plant which is resistant to the clover yellow vein virus. So, this is again a mechanism by the HDR mechanism that it can be done. So, this is the consolidated picture showing the different gene editing mechanisms where the genomes of the plants are edited. The CRISPR-Cas9 can also be used to alter the genomes of the pathogens, the viruses that are infecting. So, when the viruses are infecting, then the CRISPR, not the Cas9 basically, but the CRISPR systems can also edit these pathogen DNA so as to render them inactive and prevent the cause of diseases. And these mechanisms we have already discussed, where the insertions, deletions or the substitutions are occurring by various basic CRISPR-Cas9 as well as next generation CRISPR-Cas9 technologies to create various kinds of disease resistant plants. So, as you can see, the guide RNA and other mechanisms, they are being introduced, our desired gene targets have been introduced into the cells, and where they combine with the Cas9 systems and create all these kind of insertions or deletions. The deletions rendering the genes the disease resistant, introduction of the disease resistant genes, rendering the susceptible genes inactive, and all different mechanisms to create the generation of disease resistant plants which are there. There are other approaches to the crop quality improvement as well, which we will continue in our next session. Thank you.